Πάμε.
Ωραία. Καλησπέρα σε όλους. Σήμερα έχουμε τη χαρά να φιλοξενούμε τον κύριο Ιωάννη Παπαντωνίου, ο οποίος είναι Associate Professor στο Tissue Engineering Lab of the Skeletal Biology and Engineering Research Center, Department of Development and Regeneration, Faculty of Medicine στο Leuven, στο Βέλγιο. Είναι επίσης συνεργάτης ερευνητής εδώ στο έτος στο ΙΕΧΜΗ και έχει κάνει το διδακτορικό του στο UCL, ενώ εργάστηκε στη συνέχεια ως μεταδιδακτορικός ερευνητής στο Leuven, στο Βέλγιο. Ο τίτλος της ομιλίας του είναι Organoid-based manufacturing of biohybrid-implants for skeletal regeneration. Towards living robots? Ερώτηση. Κύριε Παπαντωνίου, σας ακούμε. Ευχαριστούμε πολύ.
Ευχαριστώ πολύ και εγώ για την πρόσκληση και ευχαριστώ και για την εισαγωγή. Βλέπετε το slide μου?
Δεν είναι στο presentation mode. Τώρα ναι, τώρα είναι εντάξει.
Τέλεια. Λοιπόν, είναι χαρά μου να παρουσιάζω στο τμήμα στο οποίο και εγώ σπούδασα τη χημική μηχανική, την επιστήμη της χημικής μηχανικής. Και πάντοτε είναι χαρά μου να βλέπω και παλιούς μου καθηγητές και συναδέλφους πλέον. Έχω ξαναπαρουσιάσει στο τμήμα, οπότε σκεφτόμουν λίγο για αυτή τη φορά να έδειχνα και λίγο κάποιες ιδέες που μπορεί να μην είχαν καλυφθεί στα προηγούμενα. Οπότε το έχω αφήσει λίγο πιο ελεύθερο το θέμα και ελπίζω να είναι ενδιαφέρον και αυτή τη φορά. Επομένως, μια μικρή εισαγωγή. Είμαι όντως αναπληρωτής καθηγητής στο KU Leuven, το Καθολικό Πανεπιστήμιο της Λουβένης, το φλαμανδόφωνο, γιατί υπάρχει και το Université Catholique de Leuven, που είναι το γαλλόφωνο και είναι 20 χιλιόμετρα παρακάτω. Είναι ένα πανεπιστήμιο το οποίο έχει, ας το πούμε, αρκετά έντονη ερευνητική παρουσία, αλλά και πολύ έντονη παρουσία στην καινοτομία και στην παραγωγή πνευματικής ιδιοκτησίας και εκμετάλλευσής της. Οπότε για σειρά ετών έχει βγει στις λίστες του Reuters το πιο innovative στην Ευρώπη και στα top 10 του κόσμου. Ενώ όπως βλέπετε το κτίριο της Πριτανίας εδώ πέρα αριστερά είναι πολύ όμορφο και είναι ένα κάστρο Γερμανών πριγκίπων του 16ου αιώνα. Στα δεξιά βλέπουμε το νοσοκομείο στο οποίο βασίζομαι, σχολή ιατρικής, φαρμακευτικής, οδοντιατρικής. Είναι ένα τεράστιο συνοθήλευμα κτηρίων, όχι τόσο όμορφο, αλλά δεν πάβει να κάνει και αυτό αρκετά καλή δουλειά. Εδώ είναι και το γραφείο μου, εκεί ψηλά στο 10ο όροφο, στον κάμπο της Φλάνδρας. Λοιπόν, ξεκινάμε μια έτσι πολύ αρχετυπική πρωτόλια εικόνα, ένα βίντεο που μας δείχνει τον πρώτο διαχωρισμό των κυτάρων σε δύο κύτταρα, η πρώτη μύτωση, σε ένα έμβριο, γονιμοποιημένο. Οπότε εδώ έχουμε τις αρχές πιθανόν ενός ανθρώπινου οργανισμού. Και βλέπουμε σιγά σιγά ότι οι αλλιεπάλληλες cell divisions, they are leading to an increase of energy. You can see the oscillations of the cytoskeleton of the cells. This increased energy will progressively give rise to the first morphogenetic event, right? And it's happening right now here. So we have a symmetric aggregate of cells that are proliferating. As they grow, suddenly something happens and they break into two different compartments, right? And this is what we just saw. So it is the differentiating morula. We have the formation of the blastocyst. So we're starting to have different cell types. We start with the same cell type, starts dividing. The first 8 cells are exactly the same, identical. Subsequently, we're going to have differentiation into more committed cell types. And as time goes by, progressively, we have more complex tissue, living tissue being formed, right? With symmetry breakage. So we have limbs growing, we have head growing, different organs growing. So this is all happening in an optimal bioprinter and bioreactor, which is our mom. And it is going to happen on and on and on until we are being born. So it is a very robust process. And that is very interesting from an engineering perspective. So we all have 2 hands, 5 fingers per hand, 2 eyes, 1 nose, apart from rare cases where we might have genetic deficiencies, mutations, etc. So evolution and development of an embryo is very robust. That is very interesting when we want to engineer things that mimic this process, right? And this is the process where all our internal organs are being generated through the emergence of 3, let's imagine them like 3 rivers, 3 basic rivers. So the endoderm, the mesoderm, and the ectoderm. And from there, we're going to have further branching into more committed, functional, related, let's say, cell types, which will consist the future organs of a human being. Now, in my case, I'm a little bit more interested into what is happening during the limb development. So I'm in a division studying skeletal tissue engineering and regeneration of skeletal defects. That is the bone, the cartilage and the bone osteochondral complex defects. So these are the type of defects we are interested in. Now, let's see what is happening here. We start having the first protrusion. So again, a symmetry breakage happening here. You see this bud popping out, and progressively what happens is that this homogeneous oval-shaped tissue will break into all this complex, limb, the fingers containing different bones. And as you see, the location of tissues within this budding tissue will define the future of the future function and future quality of these cells located in different zones. Now, yesterday, this paper came out, so I was very happy because I said, okay, it's perfect with what I want to discuss today. And what we see is embryonic limb growth, right? And they did, through 2-photon microscopy, a video, time-lapse video, and actually they mapped the velocity of cells, how is it moving in a growing tissue, right? And also we can see proliferation, real time. So what you see here is the limb growing, and the arrows are showing the velocity, how the cells are moving and how different patterns are formed within this bud that I showed you before, which looks quite homogeneous here, right? Let's see it again one more time. We see here at the center, there is a kind of compaction situation. So cells are moving from the left to right, and then there is motion downwards. And at that point, it looks like cells are being compacted. And this is very interesting. As you see here again, it's the same, only with cells that are dyed for SOX9, which is a gene that is very important for the formation of limbs, cartilage, bone, etc. And we see that there is a nucleus being formed exactly at this location where we have this compaction of the tissue. So as you see, there is now also a concept of tissue as a fluid here, right? So what we see in the next slide is that in the healthy case here, where I'm showing now, we have this green part, which is the SOX9 genes that I mentioned before. And on the periphery of that, you have cells that are red. Red means that they are proliferating, right? And they're more motile because proliferation, cells need space to move. So we have a patterning here, right? During this morphogenetic event with defined structure. And as these authors here investigated also with the use of some mathematical models, what they say is we're having a transition from a fluid state, which is actually this green part here around. What is red actually? Okay, so what the red dots are more fluidic like. And going towards the center, you have a more solid type of tissue going through the SOX9 to the CoL2. So that is very, very interesting, right? Because it shows that these different phases are modulating the way our limb is going to grow, and potentially our limb is going to break into different subdomains, maybe our fingers, right? And what you see here at the bottom side is a knockout of a very crucial gene that is controlling SOX9, and you see that this patterning is lost, and hence the capacity to grow and protrude in a systematic manner is lost. So this was just published yesterday. I found it amazing, very interesting paper from a developmental system. So it was from a mammalian system. And actually, it's a very nice way, again, to monitor in a more quantitative manner what we have been postulating for quite a long time in a more qualitative way. So why is it important for us? Because also it was on the biological system of interest of mine, and I was excited about that. And what I want to show here is that indeed what is happening also during long bone growth, we have a similar, potentially similar situation going on. So all our long bones are formed through cartilage. So what you see on the left side is a cartilaginous representation. There is no mineral phase yet. Eventually, we have the formation of these large cells. I have a section here in purple. So we have the formation of these large cells that are generated by the cells on top, cartilaginous, and they are all cartilaginous with different properties. So these cells will become more static, less fluidic, we could say. They are more rigid, and progressively they will die, go into apoptosis actually, which means it's a pre-programmed death. So it's not necrosis. So it's a way of attracting blood vessels and osteoblasts from surrounding tissue in order to make bone, like you see here. And this is the engine, this transition from resting chondrocytes, proliferating chondrocytes, pre-hypertrophic and hypertrophic chondrocytes. And as you see, there is a columnar structure also. So there is geometrically defined the way these events are happening. This is the engine that gives rise to the growth of bones. And this is why we're becoming taller and taller, right? And it is actually active also postnatally. So until we are, until teenage years finish, 16 for women, 18 for men, more or less. And this is also a process that our bones are recapitulating when they break. So long bones, when they break, they are going to go through a phase of inflammation, okay, to kind of get rid of all the damaged tissue. But directly after that, we have the formation of a cartilage tissue that again will be progressively driven towards hypertrophy, attract additional blood vessels, and then mineralize and form perfectly new bone in the place of the defect, right? And a very nice way to engineer this type of things is the use of progenitor cells, either from adult sources or the use of pluripotent stem cells, like in the case of iPS cells, what I'm showing here. And the discovery of iPS cells was quite revolutionary because it provided us for the first time the capacity to take a cell and drive it back into development. So take a fully differentiated and committed cell and form and create an undifferentiated cell. So go back in development, back in developmental time. This is what happened in 2006. That's why Mr. Yamanaka got a Nobel Prize also for this discovery. And from this embryonic stem cell, we can regenerate, guide this cell towards the cell that we would like. So we could get skin cell and maybe we can make heart cell. We can get a bone cell and maybe we can make a liver cell. So there is extreme plasticity and capacity to use this technology for engineering new tissues and implants. So actually in the plate, in the dish, in vitro, we can mimic actually to a certain extent what is happening also, as I said before, in the optimal bioreactor and bioprinter. Now, tissue engineering should make use of this knowledge in order to develop implants that can robustly regenerate defects and restore the function of damaged organs. However, for many, many years, people were trying to force cells to do tasks that they were not supposed to do. So there were a lot of random combinations of biomaterials with wrong cell types, with wrong growth factors, in arbitrary combinations that did not give rise to a lot of success. And on top of that, when the whole terminology of the European Medicine Agency came up, like advanced, this type of living products should be classified as advanced therapeutic medicinal products, there was no way to connect the quality of the tissue that we made in the lab with the process of culture, because, as I said, it was a quite random combination. Now, since that time, although since it's quite active, this trial and error approach, there is a shift towards more developmentally inspired approaches, like making use of the knowledge that is being made, even as we talk, from developmental systems. And this approach was termed as developmental engineering. And this terminology also was stemmed, let's say, also from the lab that I am heading now, from my previous, let's say, supervisor. So I am indebted to his insights also. And we're trying to push it further into more applied engineering perspective. So getting inspired by these developmental examples, but at the same time, try to make use of condensations, engineered and not single cell, random single cell solutions, condensation of cells that will have pre-programmed features. And then we need technologies that can have the precision to give us, let's say, sequence of tissues, mimicking what we showed in development, right? So we need technologies to be able to give us sequences of tissues, and we could call them maybe genetic networks or genetic circuits, or I don't know. We can have many ideas of how to name these combinations of cross-talking pre-programmed tissue populations in order to print or to produce or to manufacture tissue implants. Now, small parenthesis about skeletal defects. It's a very common injury. And normally bone has a tremendous capacity to regenerate and actually does not even induce scar. So osteoblasts are fantastic in regenerating, and osteoclasts are, balancing each other and making a perfectly new bone, indistinguishable from the previous bone. However, in cases where we have critical size defects, or in cases where we have damaged the surrounding environment, or we have diabetic patients, or smokers, or many things genetically, let's say, compromised patients, then we need to have solutions that are more potent than the body itself. At the same time, for osteochondral tissues, this is a very, very challenging implant defect that actually lies in a constant balance. So the red part here is your cartilage, and the blue part below is the bone. And these two tissues are in a homeostasis. So they are happily coexisting. However, when the balance between the two is damaged, then we have osteoarthritis coming up, and progressively we have mineralization of cartilage, and then we have a lot of pain. We have, limited mobility. We need to go and do series of surgeries, etc., etc. And this is happening more and more in young ages nowadays, also because of a highly, let's say, athletic hobbies, etc., high intensity. And like what you see here is a badly regenerated defect where cartilage is not there, and progressively it will all become bone here. So how do we make implants that can regenerate both cartilage and subchondral bone at the same time in a synchronized manner? That's the question there. So our approach is, yes, we need to move away from this random type of tissue engineering that was happening for, I would say, more than 20 years, and go back and get an inspiration from all these examples that I showed you and try to recapitulate this in the lab. So we need to make a condensation. So you see here adult progenitor cells, so we could call them adult stem cells, let's say, that have the capacity to come together and make a cell aggregate on their own, right? We see them in non-adherent wells. They are encapsulated in these wells, few cells per aggregate, and these guys come together and make a condensation. And the condensation is important because condensation is driving also morphogenesis, as I showed you before in this new paper that came out yesterday. And also through this way, what we have is a large population of aggregates that will become tissues later on, microtissues or organoids, as we call them, that have monomodal size distribution. As you see, the size distribution is very well controlled. And this means that biological events happening across this population will probably be parallel, if not identical, right? And what do we see here? Upon induction of differentiation using several growth factors, by the way, GDF5 and WNT5A that I showed in this recent publication are also growth factors that we use here to differentiate these cells. So I was happy to also see that in that paper. What we see is a progressive initial burst of proliferation. So we have proliferating chondrocytes, we can say. Subsequently, less and less of cells are proliferating. But at the same time, we have more and more of these green cells. And these green cells are these Osterix positive cells. Osterix is a marker for late cartilage, hypertrophic cartilage, the one that will give bone later on, as I was showing into this layered, let's say, picture of the growth plate of the sequence of cartilage types. So these things are happening at the same time, right? So less proliferation, more commitment to a cell that has an identity, right? And at the same time, what we see here is secretion of abundant extracellular matrix. So the dots are the cells that you see here, but this medium between the dots is collagenous-based extracellular matrix. And this extracellular matrix contains collagen type 2, which is a nice one. And actually also that was in the paper I showed before. And it was the more solid state rather than the liquid state of that tissue, right? And also we see Indian hedgehog, which is very well-known regulator for the transition from hypertrophic chondrocytes towards bone. The RNA sequencing that I show you here at the bottom shows that, in the beginning, these cells it's a biopsy, right? So we have a very diverse cell population that we get from patients. And in the first week of culture, we see huge amount of genes being upregulated and downregulated. So the green dots are statistically significant gene expression. It doesn't matter which genes they are. But what we see here is a very noisy situation in the beginning. And progressively, as time goes by, we have less and less of these differentially expressed genes being shown as statistically significant, right? And what this means, we could say, is that our biology is becoming more specific, and hence a lot of genes related to cell death or proliferation or, I don't know, contaminating cells being there and dying off eventually. And genes related to our biology might be there, preserved, and actually could be interesting for characterizing our cells as well, right? And as I showed, yes, this is again a population that is becoming more potent. And this is our new data that we did verify what we were hypothesizing before. So we did single cell RNA sequencing. This is a relatively new technology. So we can take our microtissues, dissociate them completely, and we can actually get per cell the whole genome per cell, and cluster all our cell populations based on what they are expressing. So what you see here is the initial cell population that we had in our microtissues, organoids. This is the first week, and this is the second and third week. We see that there is a big degree of overlap, right? So indeed, what we were hypothesizing before and what we were seeing with other methods is now quantitatively proven through single cell RNA sequencing here. Potentially, small differences that we might see could be also important for quality controls, etc. So potency markers, let's say, characterizing a future product. So I was very happy to see this data here. And when we implanted these microtissues as an array inside the small animal model in its back, actually ectopically, we see that these cartilaginous tissues are able to form little bones there, mineralized structures you see here in the CT. And also when we cut through them, we can see in the histologic sections that they also have bone marrow cavity, something that looks like bone marrow cavity. They have, hematopoietic relevant cell types in the center, and mature bone and osteoclasts. So bone remodeling going on. So this is amazing. Why? Because it shows that they can host the whole ecosystem for a bone organ. These microtissues that we engineer in vitro can execute the task of making bone and containing all the important cell types and functions of this organ once it's implanted. So we can call these microtissues, we named them organoids, as autonomous, pre-programmed, potentially to a certain extent, living structures, right? And what was interesting is that we also collaborated with smart biophysicists and tried to see also this hypothesis of what is going on in terms of physics. And what we saw is that when we were modulating the way that the condensation was occurring, so by modulating ROCK inhibitor, which let's say relaxes or stiffens the cell cytoskeleton, we could get a phase transition. So you see here, and that was obtained through image analysis and the mathematical model that was built, each red dot representing one cell, and being validated with hundreds of microtissues being fused over 12 hours. And we saw that in the case where we had, let's say, the good, we know that it's good because we implanted later on, but in this paper we were not doing that. But anyway, what good and bad was in this case, so what functional and non-functional organoid would be, had to do with its phase. So either it was in a more liquid phase or it was in a more jammed phase. So the liquid phase enabled also the optimal differentiation that we saw before. And that was very, very interesting because again, there seems to be some underlying physical principles governing biology also, despite its complexity and its chaotic and stochastic nature. Still, we see that when you make basic hypotheses, you can follow some principles. That was very interesting again. That corroborates a little bit again to the discussion I want to open about the living robots. Now, these microtissues can fuse with each other. They have also this capacity. They can self-assemble on their own, giving rise to larger structures. And here you see two of them doing this task. And also when we put thousands of them, like you see here, they can also make a larger tissue structure without any scaffold, so just on their own. So we use agarose wells, non-adherent wells, where we force them to assemble on their own. And when we implant them, either ectopically, as I showed here in the mouse, we have nice bone organs being formed. For the less mature, it's more mineralized cartilage. For the more mature, you see fully developed organ with cortex and trabecular structures and a bone marrow cavity. And something that does not happen when you self-assemble right from the beginning large amounts of cells. And this is not happening. You see, already due to diffusion, you have parts that are not differentiated, and this contaminating tissue that is formed in vitro is carried out in vitro, and it affects the way this tissue behaves. You see the hole here. And now, when we implant orthotopically in large defects, we see a very nice regeneration of defects, both through X-rays and also CT, as you see here. So we have bridging of a very large defect, and here it's the case after 8 weeks without any implants. Still, you have only a tiny amount of bone being formed by the host itself. And we have a bone marrow cavity or a cavity being formed. Is it bone marrow? Yes, it is. When we do the sectioning, we see that indeed the fully regenerated tissue between the dashed lines contains bone marrow, is corticalized. As you see, it's aligned with the previous tissue. It's not structurally different. And at the same time, we have a lot of brown cells, human cells. So we implanted human implant in a mouse host, and the human cells were there after 8 weeks, still contributing to this organogenesis, let's say, and these regeneration events. So this was very, very exciting data. Now, okay, let's say our hypothesis that we can indeed do developmental engineering, actually taking cells from biopsy and differentiating them in a manner that is mimicking what is happening in the growth plate, which is a very robust process. So we are mimicking a robust process. Now we want to maybe understand and measure markers that will let us know at which time point our product is ready. So we need to do further characterization of what is secreted in our medium and monitor these living robots, how they're evolving over time. Now, what I showed you before was cells from let's say, from a cell pool that we have made from different donors that we have screened. And I said, okay, now we need to become a little bit more close to reality, and we need to get different donors and do the experiment. And what you see here is 6 different donors, right? And you see one behavior here, another behavior here. Oh my God. So we just wanted to see, similar things more or less happening. But here we saw 2 distinct responses to our process. And what is even more interesting is that this is gender-related difference. So we have males on the left-hand side giving abundant extracellular matrix with collagen type 2, no collagen type 1. At the same time, the female-related organoids are much smaller, less cells, and also they do have collagen type 1. Interesting. What does it mean now? Is it this could be, we went back to the literature, we searched for papers, we found that also in mice, they have seen that when you break their bones, in females, the cartilage tissue that is formed in the defect is much smaller than the corresponding tissue in male mice. But okay, from a mouse to a human and from in vivo to in vitro, there are some, large steps taken. In any case, we said, okay, let's go and implant what we have and see what's happening. In all cases, we saw fantastic bone ossicles being formed, like you see here, with the capacity to form bone marrow in all cases, almost. We did not have a case where there was no bone marrow compartment, more or less. So that was very interesting, that despite their in vitro morphological difference, its functionality remained the same. Subsequently, we did some proteomics analysis of the secretome in order to find, let's say, generic, or how to call them, holistic, I don't know, for both genders markers that we could use in order to monitor the differentiation of our organoids non-destructively. And we did indeed manage to find some of these markers. Now we are in the process of validating them and repeating the experiment also. So we have seen our organoids up to now being evolved into autonomous structures that are regenerating a bone defect. In the next step, we decided to go for the osteochondral defect, which is very challenging. So we needed to have a layer of cartilage that would regenerate cartilage, and also a layer of cartilage that would be hypertrophic and would regenerate the subchondral bone. So we wanted a patterned type of tissue. So we developed, again, in this case, we used induced pluripotent stem cells also that are well known for having the capacity to give stable cartilage, which is notorious with adult-derived cells. And we made this dual type of pattern construct and cultured it also in a setting that would allow different media to be provided to the different sides of our organoid-based implant, as we see here. So this is the new organoids that we made from the iPS cells. You see the SOX9 and the COL2, the 2 genes that we saw also in that first paper about development. They are differentially expressed between our cartilage guys, so these ones that I show here, the blue, and the white ones that were in the previous work that gave bone. So you see the difference between cartilage tissue and hypertrophic bone-forming cartilage in regards to the tissue. At the same time, you see here the white Indian hedgehog bar is way higher for our bone-forming organoids as compared to the cartilage-forming organoids. So we do see a distinct behavior between the two populations. Although they're both cartilage, we can make different type of cartilage organoids in vitro. Although both will contain abundant collagen type 2, which is a generic cartilaginous extracellular matrix. And here you see a tissue that is, more pink, let's say, for the cartilaginous guys, or more blue for the other side. So it's a zonal structured tissue. Now, once we implant this, what we see ectopically again under the skin, what we see is that, okay, our organoids for bone, they give bone, like you see here. There is no safranino, not much cartilaginous matrix left in vivo, and a lot of osteocalcin cells. Now, the cartilage only part of the tissue gives nice cartilage. This orange part here is cartilage. And this collagen type 2 is cartilage that remains as cartilage even after implantation. And there is no osteocalcin here, as you see. And when we evaluate this zonated implant here, you see the red part and the white part. being present at the same tissue. So we implant a tissue that is zonated and it behaves differently per zone. And it's very nice to see the two zones being formed in these images. So collagen type 2 on one side, cartilaginous, osteocalcin on the other side going towards bone. And here's some zoom ins of this zonation.
But interested in
So,
the melt electrowriting.
Sorry.
Since it's a solvent-free product.
So now, how do we scale up this type of implants? Because it's all nice, we are able to play around with mice, as I showed, but what's happening when we want to go to larger implants? And being able to form like fully filled organoid-based structures, it's very difficult and very expensive to produce this type of volumes. So we decided to merge our organoids with mesh electrowritten, with melt electrowritten meshes in order to make biohybrid type of membranes that could be foldable, that could be handled by surgeons, etc. So what you see here is also this mesh that we have seeded with these organoids. The dimensions of the pores are similar to our organoids, so we were hoping that they will be entrapped. And at the end, we make this very, very fine, membrane that contains our organoids in also a 2D plus manner. When we implant, you see that again, we get a bone organoid here. And also in the bone, you see some of these lines of the mesh being present there, which is very interesting. At the same time, this methodology, melt electrowriting, also provides very low volumetric presence of a foreign material in our tissue, which is something that we also want to maintain for not having, let's say, host versus graft responses, etc. Now, how do we culture them in suspension? We are engineers also, and I thought this is also interesting. So we developed this little bioreactor system that can, run parallelized batches of cultures. And so what we saw is that when we have low RPMs, we have a clustering and agglomeration that is uncontrolled, leading to large formation of tissues that we don't want, so it's not controlled. We could find a regime where we could have a suspension, and when we went to very high RPMs, we would have destruction of our microtissues and the formation of debris. And at the same time, we also had some, because they're quite sticky, these microtissues, we had some controlled fusion also during, let's say, the optimal window of operation, as you see here, especially during the initial phases of culture, something that was, let's say, phased off later on. And we tried to also develop a tool that could allow to characterize the stress environment that each of our organoids would experience, because this is we cannot assume a continuum here, right? These are quite big structures floating around. It's not very dense. So there would be no sense in just saying, okay, we will do, finite element analysis of the liquid and these shear stresses correspond to what our microtissues actually see. So we coupled an agent-based model with a continuum model in order to be able to explore what type of shear stresses our population is seeing. And what you see here is also the distribution from the media. And in low ones, because we have also this formation of the pellet, you see a very large, let's say, distribution, especially towards the lower amount of shear stresses, something that phases off later on. And we kind of defined also critical amount of stress that the organoid needs to see. As you see here, for example, what we get is interrogation per organoid of its shear history, shear experience, and also the collisions that are happening between our organoids. Now, what is interesting is that we also saw that when we found the optimal condition of operation, we had a very low amount of dead cells, so comparable to what we had in the static condition, with less proliferating cells. So the provision of mechanical stress in the bioreactor, as you see here, results in less proliferation, but at the same time, more extracellular matrix. So at the same time, at the gene level, we see commitment to hypertrophy, CoL10. This is the bioreactor, CoL10, the bioreactor. Indian hedgehog, we have, as I showed before, much higher upregulation of these genes, the same for chondromodulin. And also because we have a system that is mechanoresponsive, right? It's the skeletal system that we're exploring. So this system could potentially be used also at a larger scale. Now we have a very small scale here to produce potentially clinically relevant amount. And also we carried out some exometabolomics analysis in order to be able to, again, define some markers that we could potentially use to monitor and assess the differentiation stage of our culture. In both cases, starting and bioreactor gave again bones. Here is a brief section of this. So the potency was retained. So now how do we make, we saw a lot of, suspension cultures, how they behave on their own, self-assemblies. How do we engineer them? How can we engineer now living tissues that have pre-programmed properties, and hence we can call them a little bit of robots, since they have the functionality now that we can control to a certain extent. So organ printing through spheroids is quite old. You see, it's since 2009 has been suggested, but to be honest, it's only the last 2-3 years that we see actual implementation of this approach. And in the traditional way of bioprinting, we had a lot of this, we're going to print something that looks like an ear, something that looks like a bone. However, it does not function as a bone and it does not function as an ear. So we have, as you saw, we had to go back to development to get inspiration and start engineering now living media that can be used to engineer implants. And here you see some of quite recent publications where different groups, leading groups, have used robotics to start forming organoid structures that are more complex, so aspiration and deposition of organoids, or extrusion printing through a slurry, so a dense medium of organoids. These are things that are very important for us, and we are also exploring it and studying in the lab. So again, you see here a robot that can actually, through image-based detection and targeting, aspire targeted organoids, as you see here in the video, and release them and deposit them at predefined locations. Also, we were able to make implants, larger implants, that we saw again that we explored a little bit where the diffusion challenges would be during differentiation for this specific biology, something that was not very well known before. Now, we also explore bioprinting technologies besides these robotics-driven approaches, where we can target and print single organoids. In this instance, we're exploring laser-assisted bioprinting. So we can have a medium with our aggregates, we can image them, target, there is a jet that is formed, and the aggregate is flying, and we can make this array of organoid, let's say, based droplets that are alive, as you see here, and also they are able to differentiate with comparably to the static, the non-printed ones. So the method seems to be matching with the needs of what we have. And here you will see again, targeting of an organoid, and laser will hit it and transfer it into the well that is below in the substrate. So it is a method that allows us to build properties in future tissues inside out. So we can build in properties. So this is very much matching the quality by design principles. We are also exploring extrusion bioprinting, as you see here, for the formation of more conventional scaffolds containing cells. Also collaborating with George Pasparakis from Department of Chemical Engineering, we're exploring functionalized alginate-based bioinks, thermoresponsive. So how can we print and how can we use this thermoresponsiveness to release organoid structures or cellular structures? And the idea now is, how do we make a pipeline that we can engineer these implants? So as you saw, small scale, it all is nice. However, when we want to go to the clinics, we really need to have a systematic approach where we have scalability for the expansion phase, so we have enough cells, robotics to support our manipulations and standardize these procedures, and also the design and the bioprinting of technologies that are, however, matching the needs of organoids and not single cells should be developed. So sorry for this. So we have this Horizon grant here that is ongoing. Also, I'm coordinating this through KU Leuven, but also the fourth institute is participating through Maria Klapa, also for characterization metabolomic characterization. What we want to build here is a new, let's say, factory, let's say, for the production of skeletal implants, organoid-based skeletal implants, like the ones I showed you before, but at scale. And so we're going to have a unit that hosts bioreactor to culture our organoids, a robot that will be able to sample and take samples for quality control during culture. We're going to have centrifuges, microscopes embedded, so all this will be fully automated. At the same time, we'll have a bioprinting setup with portals that can open, close, and maintain GMP. So here we're moving a little bit towards the GMP manufacturing, so we have to comply with this type of regulations. And hopefully by the end of 2-week, 3-week period, we're going to have a functional implant that will be able to heal defects in large animals, in this case, mini pigs, and in the future, humans. Now, going towards the end of my presentation, there was last year this hype during also the COVID, the pandemic and the quarantine, was this explosion of the Xenobot. Maybe you saw it in the news or it was broadcast by CNN, BBC, Greek channels, etc. So some researchers developed an algorithm that would predict how cells should self-organize and function, right? And here is the algorithm, and here is the result of this prediction. And also you see other type of predictions in other settings. So what is this about? I quickly will show you a video. So here you see these robots, as they call them, the Xenobots, going around single cell suspensions. And they are forming some kind of aggregate, like I showed you before. The shape of this Xenobot was designed like this, as you see here. And its function is that it not only moves as predicted, but it can also form daughter structures that behave similarly. Now, okay, sounds very exciting. These are novel living machines, as they claim. They're neither a traditional robot nor a known species of animal. It's a new class of artifact, a living, programmable organism. Potentially, as you saw, there are underlying mechanisms that we are discovering slowly. Potentially, we are going towards that direction. I think this was a little bit overhyped. Το τραβήξαν από τα μαλλιά, που λέμε και στα ελληνικά. However, it's a very thought-provoking type of work. And also, previous one on this direction again was the engineering of Stingray with designed features in its exoskeleton, and using cells as an engine to drive its motion, as you see here. So it could swim based on the balance between the forces exerted by the cells and the exoskeleton that was embedded in this hydrogel structure. Now, what we do on that front, we are also interested in this. So how can we make a cartilage robot? So how can we break the symmetry in order to see some morphogenesis in vitro? So what I showed you before was always spherical type of organoids. But now we're trying here, collaborating also with Maastricht University to make maybe environments that will induce this breakage of symmetry. And these are very recent also data I would like to share. And like you see here, the yellow part is extracellular matrix, collagen, but also you see another part on the other side with a lot of cells being present that is not yellow, so it does not secrete collagen, you see again here. And this to me reminds a little bit of what we saw in that first paper that I showed you, that you have a more fluidic phase. Potentially we see this here also on our right-hand side, and a more solid and collagen containing the collagen type 2 phase on our left-hand side. So is it something that is an artifact? Is it something that is true? We need to do more repetitions. But for me, it was a very nice result to share with you today here. And also, what we're exploring in order to make this type of robots is also how do we control it? Because with robots, you control them also externally with certain means. So we are trying to also collaborate with University College London to embed magnetic nanoparticles in our structures in order to be able to remotely also provide signals to them. So not only engineer it through our media and our agglomeration, control of the agglomeration process, etc., but also externally. And here you see a lot of organoids inside the hydrogel. And what you can see in this case is it's not a magnetically actuated one, But here, when we apply magnetic force, you see a lot of linear structures. So the cells are starting to grow into giving tissues that are more, let's say, polarized structures. And that was quite interesting to see. And in addition, what we can use it, as you see here, these are our tissues, and upon magnetic, let's say, actuation, they are coming up here. And also when we make some patterns with the magnets, our modules here, our tissues will kind of take the same designs that we're giving. So we could maybe elaborate the design of magnetic fields that can guide this type of processes. And what you see here in the video also is, external modulation and movement of these tissues. So in our brain, potentially in the future, maybe we can accelerate bone healing of magnetically augmented implants, because again, as I said, these provide mechanical stimulation to our implants, right? And final slide, we can also engineer genetic networks. So we have CRISPR-Cas technologies by now that we can actually go in the cell and actually activate or deactivate specific genes. As I showed you, SOX9 is a very important transcription factor controlling this cartilaginous patterning and differentiation. So we could also go into the software of the cells and start making the cells more predictive in the way they're behaving. We could provide, as I showed, a better platform to culture them as an organoid, and also we can actuate them from around through different means without actively handling them. So taken together, if we take all these things, potentially in the future, we can have an algorithm or a model that can say to us, okay, I need 1 centimeter of this tissue, half a centimeter of that tissue, and the vasculature should be so dense in order to have a response. So that is the dream. That's where we are trying to go to, let's say, in my case, for the skeletal system. I would like to thank Professor Frank Leuten, who was my mentor, let's say, and he taught me a lot of things. Also, I would like to thank my current team at KU Leuven, and also the people I'm collaborating at for the students and the postdocs. So I would like also to thank the funding bodies that are supporting all this research that I showed you these years. And finally, I would like to thank you for your attention, your patience, and I'm looking forward for some questions if we have time. Thank you very much.
Ευχαριστούμε πάρα πολύ, κύριε Παπαντωνίου.